Hello students, welcome again for the biology lecture. So last lecture we have discussed about uh, synthesis of mRNA, and in the synthesis of mRNA, that is the message is given from DNA to the mRNA, which type of the protein is to be synthesized. And as per the need in a eukaryote special, there are some processes takes place or occurred in the mRNA before starting of the protein synthesis. So the protein synthesis, which type of protein is to be synthesized, it is written the message on the mRNA, and for that purpose, there are different steps involved, and those are known as the processes for the further uh, formation of mRNA, and those are called as different names. That means those are capping, then the uh, tailing, capping, tailing, and splicing. So capping is the addition. Capping is the addition uh, of different things which are required for the process, and then there is a addition. There is a addition of in a capping. There is a addition of methylated guanosine triphosphate, and in tailing there is a addition of one more thing, that is the polyadenylation, and in addition to this there is a deletion, or the which are not required for the genetic information, that is the intron, which is a dark site of the segment on which is written on the mRNA, that is cut and removed, and so that the intron which are necessary, exon which are necessary for further process where the genetic information is given, and only exon they are jointed with one another, and intron that is cut, or that is totally removed. So that process is called as the splicing. So in this way, there are three processes before the starting of the protein synthesis. So we have discussed so far from uh, in the last lecture. Now let us start with the next part. Before protein synthesis, we must know some things, and that things that we are going to discuss. The first one, that is the um, characteristic of genetic code. Characteristic of genetic code, which are very interesting, and these characteristics we must know before the starting of the protein synthesis. Okay, so let us start with this. uh, so share some ppt. So this is the slide, first slide, and which having what are the information, that is the characteristics of the genetic code, those are given here. So let us see and discuss one by one. So characteristics of genetic code. This is the first characteristic, where it is the genetic code is in a triplet code, and there are three consecutive bases. That means three purines or the pyrimidines. These are the nitrogen bases, and they are arranged in a triplet form, which are called as a code. So one code that is represent uh, for the specific type of the amino acid, and many many codes maybe represent the single or many type of the amino acids, but this is a triplet code, and that is written in three nitrogen bases, and they are called as the consecutive bases, which are read only in the 5-3 direction. That means if we go to opposite direction, the meaning of amino acid that changes. Okay, so uh, let us see the next one. Here, the genetic code has a distinct polarity. What I said before, that it has a specific type of the polarity. It should be read only from the one direction, and it is always in the 5-3 direction, not 3-5 direction. If we go to the For example, AUG. AUG is the methanoiodine. So suppose this is a AUG is methionine, and if you go to in opposite direction, that is a 3,5 direction. So instead of the AUG, that is a GUA. So it changes the meaning, and hence it changes the amino acid, and hence such a type of the reading it should be only of from the 5-3 direction, not opposite direction. Hence, this genetic code has a distinct polarity. You must remember that when the polarity changes, or if we read in a opposite direction, then the meaning of that code, triplet code, also change. That changes the amino acid. Next thing is that genetic code, which is written in 3 nitrogen bases, that is non-overlapping. Means what? If the consecutive or serially 3 nitrogen bases are arranged, so only first three that represent one meaning, that is the genetic code, but other three that represent another genetic code. So these codons, they are not overlapping. So genetic code is never overlapping, though they are arranged without any gaps or comma or whatever it may be, but they are non-overlapping. Overlapping is not possible. That means we can say that it does not obey any type of the punctuation or grammar. That is without full stop or without comma, like that. So no any type of the punctuation, just like grammar in English. So it does not obey any but it is a comma-less. So continuously it is arranged without any gap, full stop, or the comma. So it is a genetic code. It has its own characteristics of genetic code. Next thing is that about genetic code has a degeneracy. Now degeneracy means what? Degeneracy. These genetic code are arranged and single triplet codon, triple code that represent the single amino acid, or the amino acid may be represented by many triplet codon. So there is no degeneracy, and hence this rule that having, that is obey strictly, that the single, it may have the single triplet code may represent single amino acid, or many triplet codon represent uh, different types of, or the same uh, single type of amino acid. In short, single triplet code may represent single amino acid, or the amino acid may be represented by many triplet codons, and hence it is a degeneracy. Degeneracy can be expressed by, or more explained by, oval hypothesis, in which first two nitrogen bases, those are confirmed. That means they are particular. But the third nitrogen base is oval. That means it is not fixed. It may changes. But the identification from the anticodon, which is written on, which is present on the tRNA, that recognize. So the first two nitrogen bases are important. Third one may be variable. So this is called as the degeneracy. This is all about the Obel hypothesis. Obel has explained these things, that the third nitrogen base is not a specific, or it is not a, particular type. It may change. It is not of the complementary type. But the first two, those are complementary, and it must be there. Then and then only the complementary can be recognized or identified by the tRNA. So in this way, degeneracy is there. Next is about the genetic code is universal. Genetic code is universal because in all living organisms, if you see, these amino acids are particular, they are specific. That means in all over the world, whether the living organisms that live, maybe aquatic or the land habitat, doesn't matter, but all having the same type of the codon or triplet codon that specifies particular type of the amino acid. and hence it is a universal. That means it is accepted universally, and hence this acceptance is not change. It is not change for different types of the organisms, but from the bacteria to the largest type of the animals or the plants, it is same one. So the genetic code that specifies specific type of the amino acid, it is same. And we know that in a living organism, if you see, there are about 20 amino acids are there, and this 20 amino acids, by combinations, that the permutation and combination, they represent different types of the protein or the amino acids, but they are universal. That is the same name is there for microorganism or the giant organism. So it is not changed. So it is a universal. Next thing is that about genetic code is non-ambiguous. Genetic code is not ambiguous. It is meaning what? The specific amino acid is encoded by a particular codon. So particular type or the specific type of the amino acid is coded by a particular codon, and alternatively, two different amino acids never encoded by the same codon. That means they are specific, no change, and hence these two different amino acids, they are not encoded by the same type of the genetic code or the codon, and hence this is non-ambiguous. So specific amino acid is encoded by a particular codon. So these two amino acids never coded with the same type of the codon, and hence it is a non-ambiguous. Next thing is that initiation codon and termination codon. If you see in the mRNA, what is written on the mRNA, and during the protein synthesis, this is a very important thing, important characteristics, that there are two ends which starts from the 5 and ends with the 3. So it is a specific in all living organisms. If you see, it is the polarity is there, that is a 5 3, and the codon also they read in same direction, that is the 5 3 direction, not 3 5 direction. What I told before, that if you read with the opposite direction, the codon meaning changes, as well as amino acid also changes, and protein is also changes. So to keep the same meaning, this polarity is very specific, and it is 5 3 direction. Okay, so at the 5 direction written, that is on the mRNA, generally it is the codon known as the initiation codon. What is the meaning of initiation codon? Initiation codon means here the protein synthesis starts with this type of the codon, and it is with AUG. AUG, adenine, then uracil, and guanine type. AUG, so it is called as the initiation codon. For the initiation codon, there is a specific tRNA is there, and AUG represent as a particular type of the amino acid, that is the methionine, which is a specific known as the initiation codon, where the protein synthesis starts only with the AUG, not any other type of the codon. Okay, at the second end, so this is about the starting. Now at the last, at the three end, there is a termination codon. Now this termination codon is also particular, but it is a meaningless. So such a type of the termination codon, it is with no any meaning. That means it does not represent any specific amino acid. So this codon, it is uh, known as meaningless. It is known as a meaningless, or it is a termination codon. It does not represent any type of the amino acid, and it generally having three codons are there. Termination codon, initiation are only one, but the termination codon are three, and they always start with the UAA, or UAG, or it is the UGA. So uh, at a time three may be there, or two, or single, but it represents the termination codon, and there is no specification that is the anticodon that present on the tRNA, and hence this termination codon is a senseless. It is called as the senseless codon, because it does not gives any sense or any type of the amino acid, and hence the 5 3 codon, or the 5 3 direction on the written on the mRNA, messenger RNA, it always starts with the initiation and it ends with the termination codon. So this termination codon, that if it is there, that is UAA, UAG or UGA. If it is present, then it is also known as a stop codon. That means it provides the message that protein synthesis must be stopped. So this is the end type of the mRNA. So that is the three end where we have to stop. That means the protein synthesis is must stop, and it does not represent any meaning to this codon, or it does not specify any amino acid. So this particular 5-3 direction, that having the two types of the codon at the both the ends. First is the initiation and second is the termination. So initiation, that indicates starting of the protein synthesis, while termination indicates the stop of the protein synthesis process. So in this way, this is the important characteristic that is written on the mRNA as a genetic code. Next is that universal. So this usually in all organisms, what I said before, it is usually in all living organisms. It's maybe microbes or the having bacterial like the cell, or giant type of the cell. See, this codon specifies the specific amino acid. So this is a universal. This is a universal. That means there is no change in the amino acid, the sequence that is from the 5 to 3, and it specifies a particular type of the amino acid. So these amino acids, they are arranged, they are linked with one another, polypeptide bonds, and this polypeptide chain is there, that is indicates particular type of the protein. So these proteins, those are different. For example, in plants, if you see the proteins are different one. In animal, again the proteins are different one. And there are so many types of the different proteins are present, and those proteins are synthesized as per controlled and governed by the DNA. And hence, this particular type of the protein is only present in a particular type of the organism. But this specification of the codon that specify amino acid is very particular, and that is universal. That means one type of the amino acid, the same meaning is there for the amino acid, the same type of the amino acid present in the another animal or the plant, but having the same name. So it is a universal. In this way, this is the ninth characteristic of genetic code. And the last one, that is the codon and anticodon. Now you can see here codon. Codons are, it is a part of DNA. What is codon? It is a part of DNA which is present mainly on the template codon. You can say template strand of the DNA. And this codon are transferred, but they are complementary. Complementary codons, those are transferred. According to that, they are written on the mRNA, but the codons are present on the DNA. And if you see the again the polarity is there, that codons always having for example AUG, methionine again. So it is a 5 AUG and 3. So this can be read only in this direction, 5-3 direction AUG. So this is example of the codon. Now anticodon, it is a part of tRNA. Anticodon is a part of tRNA. Means these anticodons are complementary with the codon. Anticodons are complementary with the codon. For example, we know that A always pair with the thymine, but instead of this in DNA, thymine is replaced by the uracil. So A always pair with the U, adenine with the uracil. It is by the double bond formation. Okay, so A and U, those are complementary to one another, and this uh, pairing of another purine and pyrimidine, that is the C and G, that is cytosine and the guanine. So in this way, these are this is the normal pairing of complementary nitrogen bases. Okay, but the anticodons, they are complementary means what? Anticodons are written, or they are the part of tRNA. So at the base of tRNA, if you see the model of hairpin model or the cloverleaf model of the tRNA, at the base of this, the arm, there are anticodons are there, and they are of course triplet form. They are the triplet forms, and this anticodon are complementary with the codon, and hence what is the message that is given on the mRNA? According to that, this triplet codon on the mRNA are complementary with the tRNA. That means on the tRNA, at the base of tRNA, it may be of the clover leaf or the hairpin model. This triplet codons are written. Those are complementary with the codon which is written on the mRNA. So in this way, such a type of uh, characteristics of gene genetic code is there, and we must know before the starting of protein synthesis. So this is all about the characteristic of genetic code. We will uh, see again in a brief what are these characteristic, because this is important with the point of view of exam. So there may be question on the characteristic of genetic code. It may be for 3 marks. So you must know about the at least 6 points or the characteristics of genetic code. So let us see again in briefly. The first one, that is the genetic code, is a triplet code. That means it is written in the three nitrogen bases are present, and they are consecutive 3 bases are there in a 5-3 direction. Second characteristic is the genetic code has distinct polarity. That means they have two poles, 5 and 3. So they are always read. They are always readable only with the 5-3 direction, not opposite direction. If you read with the opposite direction, then the meaning of this codon changes, and hence the different type of the amino acids may represent. But you must know about that it should be read only and only from 5 3 direction. Then third characteristic is the genetic code is non-overlapping. So what is the meaning? If consecutive 6 amino acid, triplet codons are written, even though there is no gap, no full stop, but they are non-overlapping. That means 3 means only 3 triplet codon. First 3 codon represent particular type of the amino acid, and the fourth should not enter inside, but it is a non-overlapping, and hence triplet codons are only triplet forms, not the fourth or fifth or the sixth amino acid. Nitrogen basis is added, but it is a non-overlapping. Next character is it is a commaless. So no any grammatical rules are there. Uh, though it is written in a continuous formation format, that without any gaps, without any space, without any full stop or the comma, and hence this genetic code, they are only three types. So it is a triplet and commaless. Then the genetic code has a degeneracy. That means single amino acid that represent single. It is encoded by single triplet codon, and if there is a next one, that means it is not readable, but this genetic code that represent single amino acid, or in some cases that another amino acid may be indicates or coded by many 1 2 3 type of the triplet codon. So there is a degeneracy, because we know that there are about 64 total codons are there. Out of that, 3 are senseless. So only 61 codons are there, and they have the meaning. So there is a degeneracy. Next is that genetic code is universal. What I told before, that this having, it may be microbes or the giant type of, so unicellular or multicellular doesn't matter, but the meaning, that the genetic code represent that particular type of the amino acid, and it is specific. It is all over the universe. So it is a universal. Next is the non-ambiguous. That means what? Single amino acid represent maybe by single triplet codon, or another amino acid may be uh, encoded with many triplet codons. So this is ambiguous. It is specific. In short, it is specific. Particular type of amino acid represent biparticular triplet codon. So there is no any change. That means uh, the same amino acid cannot be recognized or coded by different types of the triplet codon, but it is a specific. Then the last one, that is the initiation codon and termination codon. What I told before, that initiation AUG is there, and it indicates the process of protein synthesis is continue or it is start, and it is generally AUG. While it also represent uh, just one amino acid, that is the methionine, and the last one termination codon, it is maybe of three types of the codons, triplet codons are there, which start with the U, that is the UAA, UAG, or UGA, and this termination codon does not represent any type of the amino acid, but it is meaningless. But only it gives the information that you have to stop the protein synthesis. So it is a termination or stop protein synthesis process. So it is present on the mRNA messenger RNA starting from 5 to 3 end. So at the 5 end or the initial end, it is the initiation codon AUG, and at the 3 end there is a termination codon. So 3 or out of 3, 1 or 2 may be there. Then this is a universal, what I told before, that the specific amino acid that is specific, and it is the same to that of the other uh, organisms. It may be different types of the organisms, but they represent the same type of the amino acid in all over the world, all over the living organisms. And the last one, that is the codon and anticodon. So here codons are the part of on what you can say, that is on DNA. It is a part of DNA and it is a codon. While anticodon is a part of tRNA and it is complementary with the codon and it is present on tRNA. So in this way, these are the all characteristics of genetic code. You must know about characteristics before starting of the protein synthesis. Okay, so let us stop here and we will discuss again some things are important for before we start protein synthesis. So we must say we must talk, or we should know about the knowledge about the tRNA, how it is act as adapter, as well as the different uh, things that is which are required for the protein synthesis, which are the raw material. We must know all these things before starting of the protein synthesis. So let us stop here and we will meet again for the next lecture for the protein synthesis.